gentlemen, I've got the absolute pleasure of introducing to you Jason Williams, which for me is an inspiration. And he's one of the leading voices in bioinformatics education worldwide. And he's got a lot to say, not just about the science itself, but also about how to bring and how to make it more accessible to disadvantaged communities from around the world, which to me is one of my greatest passions. So thank you for joining us, and it's lovely to have you with us.
All right, wonderful. Let's see if I can make this full screen. Okay. Oh, good. Everyone can see. So thank you for this. I should, like I said, like a cup of coffee. I just sent a message on WhatsApp because I've been trying to catch up with Noel for some time and just see how things are going because it's been a little while since we've chatted. And it's like, hey, maybe Friday I can have a cup of coffee with you. And then it became a lecture. So I'm very happy to do that. I was just visiting, as I said, at a meeting that was in Cardiff, and I was around this week, so I'm happy to do it. We have a nice audience here, so I hope that we have actually some engagement in talking. But I guess the value of the slides is I can show you some pictures and show you things that I'm working on, but I really hope to have a chance to listen and learn from you. So I'm at Cold Spring Harbor Laboratory, New York. Actually, today is my 20th anniversary.
Oh,
I
wow.
started 20, yeah, I'm not there. And exactly, right.
Does it actually look like that? Or you
Yeah, no, that's a painting from across. We're on the North Shore of Long Island. I grew up actually a little bit south. Yeah, some people think when they're driving, even people from Long Island think it's like a bed and breakfast. But it's actually a research laboratory that specializes in cancer, neuroscience, quantitative biology. Probably where I'm standing on the map of Cold Spring Harbor in the photo is probably where Jim Watson's house still is. So he still lives on campus. But I'm within a particular unit, which is the DNA Learning Center. And so this was started in the late 1980s as a way, it was the first education center that was devoted really to biology, molecular biology, genetics, bioinformatics, starting at those secondary levels. And as Manny already introduced, I work on a number of different projects there, and I'll get a chance to tell you about some of them because my slides were hastily assembled. It's probably too many slides I have, but I tried to focus on some things. So the Learning Center sees tens of thousands of students itself, and then our products or our partners and things that have sort of been inspired by that. We think it's probably on average around 500,000 students touch some of the materials that we've been involved with. And I'll mention some of them. As I said, we do lots of hands-on education in molecular biology starting in middle school. So really the first labs with transforming bacteria and learning about the cell. These are enriched things that some school districts will have, but a lot of school districts actually don't. And so the fact that they can visit us to supplement their education and make a really fun hands-on experience. We don't give long lectures, we just do stuff. Our high school programs continue that with some experiments that are aligned with the curriculum, but a little bit more advanced. We have a number of project-based explorations I'll mention something about, but some of those involve mentoring. So because students might only come to us for a few hours for a field trip, that's not enough time to really get to know them. So we have other programs where we meet with them on a regular basis. So even last night at 8 p.m., I just tuned in to have a monthly update with some of the students who are in one of our programs, and we just check with them. In that case, these are students often from minority or disadvantaged populations, and in that case, we're helping them, okay, are your college applications done? Send us your essays. Here are strategies for asking and collating letters of recommendation. So we really work with the students intensively. I spent probably the majority of my training, my time on faculty training. What I guess I'll try to talk about today is really how do we prepare faculty who are at institutions serving undergraduates to enrich what they're doing so that they can bring it directly to their undergraduate students. So we spend a lot of time with that, online education. And then in our location, the Dolan DNA Learning Center is the one that's right in Cold Spring Harbor, where my office is at. We have 2 locations in New York City, one up in Manhattan, Harlem, which is embedded in a school complex there. We have on the lower left-hand side, DNALC NYC, which is in Brooklyn. It's literally right off at the foot of the Brooklyn Bridge. So as soon as you leave it, you can go walk across the bridge when guests come. But that's a whole floor in the City University of New York. And we have 6 laboratory classrooms and 2 computer labs and prep labs and internship program, etc. We also partnered with Regeneron Pharmaceuticals. So they are very interested in training and outreach. Actually, their CEO is basically grew up on Long Island, where there's an atmosphere of students doing science fairs and doing all sorts of talent things. So they sponsor a national or international science talent search. But then they also realize that getting students at the earliest possible age to do science is really, really important. So across the world, there are a number of centers that we either have inspired or run together or co-licensed. So the largest one is in Suzhou, China. The newest one outside the US is in Nigeria. And then we're working on one, it's just how many. The next one will probably be in Tennessee at the Meharry Medical College. Is one of 4 historically black schools that have a medical college. So I'm very happy and privileged to be part of a very small group that has an international influence on this really early exposure to biology education. So sort of my background, I guess part of my background is definitely about computation and the fact that one of the things that gets left out of the mix, because we do a good job of getting microscopes in front of kids, is also getting them to think about data and has something to do with it. So I think a lot of what my goal is, my job description is really to help researchers and educators learn how to use data and computation effectively. I think maybe we're doing better, but we still, I just taught at one of our advanced sequencing courses, was teaching some introductory Python. And it's making its way into the curriculum, but it's still a very valuable set. It's also changing, right, because AI is here. And so what we have to teach, or maybe what we can skip teaching, because actually you don't need to know that much and you can let the AI do it for you. Or maybe I need to focus on basic principles so that you can focus on what you want. Those are all open questions. But undoubtedly, the skill set is changing. If you do I did this graph some time ago, it's probably still true, of just searching for any publication that mentions machine learning, and it's not just that the absolute number of publications making use is high, it's that the rapidity, it's such a rapid increase. Unless you're basically going to say as a biologist that I'm going to take a year off to learn this, there's no way that you're going to keep up with it. You hope that you can get this in front of your students. You hope you can pick up pieces of it. But essentially, and to quote a paper, which I like so the title is not my title. It's a title, actually, these are people who work in machine learning, saying it's very difficult to keep up. So clearly, we need better strategies. Because we're not just going to, at least not in the immediate future, plug ourselves into a computer and learn overnight or something like that. We need different strategies, right? Because our current strategy, if we think about the skill set to be a molecular biologist, which is my training, depending on what decade, right, there were really core techniques. But even though those core techniques changed from time to time, they're all related, right? I have done absolutely nothing in a lab with CRISPR, but if you put me into a lab and give me a pipette, probably in a day or two, I'll get something working. But when it comes to the computational skill set, you started off saying, well, somebody needs to know how to have typing skills to be a biologist, because maybe you need to type up your paper or write a grant, to saying that you need to learn how to use AWS to configure a couple of servers and launch something. That's not the same level. It's not the same request. So that's a big skill gap. On top of that, we all know that the data are huge. This needs to be updated to see if it's accurate. But this is a paper from some time ago by at least one or two colleagues talking about that you shouldn't say astronomical to mean big. You should say genomical to mean big. Because the amount of data that's out there for people to think about is really interesting. As educators, I really love this quote. This is from Economist, looking at changing skill sets. And we always talk about, and probably every single politician who mentions science mentions, well, not in the US recently, but every politician usually mentions science, says something along the lines of we need more scientists, we need more STEM researchers and skills. But what it turns out in this particular analysis, it's not even so much that you need more people with STEM degrees. It's recognizing that we need better ways to keep people who have a STEM background updated and current. And what they found in that particular analysis is that you're graduating STEM graduates, but then they reach a level in their career which they're not able to get more training. And they just sort of either just have to decide that I'm happy where I'm at, or they have to exit the field because they're not really good ways for them to come in, right? So you might have spent a good amount of your training learning how to sequence a genome, let's say. But then all of a sudden, like happening now, oh, you need to know how to use AI to analyze that genome. And if you can't do that, then you're kind of stuck with a skill that somebody is looking less and less for. And it's something that we need to think about. That said, genomics as a framework or as a vehicle, obviously I'm biased, so I'll admit that, as a vehicle for education is really interesting. I think about it like this. Every single person, every single culture, you usually have a document or an artwork if you want to extend it, or music even, you could think of as document if you're thinking in philosophical terms, where instantly you see it, you recognize it as being part of your culture and part of your society. All of these documents, you might, one or more of them, you might look at them and say, I relate to that in some way, or it reminds me of something that I connect to. But we all have a genome. We all have the same 99.93% of letters inside of us that make us completely unified as a species, right? And so it's this thing that we all share, but none of us know anything about. When we look at chromosomes, you probably don't look necessarily, some people do, with affinity and say, that's what makes me human, and you have the same ones too, and therefore we're unified in that way. But we should, right? Because if we can get students thinking about genomics, it can touch a lot of subjects. The history goes back, right? This is actually at Cold Spring Harbor. This is in 1989 at one of the first meetings to organize the sequencing of the human genome, right? And there are people that you can see there then who started the process and then went off to achieve it, become NIH directors, continue in other important discoveries. But this is a really recent, right, timeline of the major points and milestones that led to us getting to where we are. And to make a comparison, right, about our ability to understand the genome, the first telescope was actually Lippershey, which Galileo based his scope on, but that's at 1608. And at the same time, he's probably taking observations with, something like a pen and paper, right, pencil and paper, parchment. And it took 400 years to go from roughly the same, ground glass lenses as a telescope technology to something that could really let you see the universe. And I just happened to choose Kepler, so I get a round number, 400 years. And hard disks, because now you're dealing with large data sets. But with human genomics, it took about 40 years to go to that, trying to convince you, same level, right? The first sequencing was actually RNA, and that's in the late 60s. Still writing probably with pencil and paper, actually a ruler to measure the bands on the gel. And 40 years going to the first machines that allowed you to do high throughput sequencing and also use hard disks. So that's like somebody's career, right? And the technology, as we know, keeps accelerating. So, Nanopore, which is my new favorite toy, allows us to do this with a handheld device, and I'll talk more about that. So we've gone from this, and at the same time, the other paradigm shift is that in other areas of science, again, my thesis here is that genomics is really interesting in the opportunity for science. In areas like science, like physics or astronomy, their devices are becoming bigger and more centralized, expensive. You're probably not, there's some parts you can get the data in front of the students, but you're not necessarily going to have an undergraduate operating a telescope or having access to CERN. Biology did exactly the reverse, which is at one point you'd go in genomics and you go to a conference and they would have a map, and the map would change every time you go to that conference, where they pinpointed who had a genome sequencing device at their university, and the numbers would go up. There's no such map anymore, because literally every single person can have a genome sequencer sitting in their desk. And this recent transformation is what allows the price of genome sequencing to drop more than 50,000-fold. Really, it's 100,000-fold, from something that's costing $10 million per megabase in 2000, and now much, much less. I always, with my 10th grade students, although now they're getting older, which means I'm getting older, but I always ask them, where are you on that map? And they're kind of catching up with this. But I'd like to point out, it's like, yeah, when you were born, if there was an issue, if there was some illness, if there was something wrong and the doctors needed to sequence your genome, it would, how many of your parents would pay $100 million? All right, so it wouldn't be possible. But now for us to say that. But equity is still an issue, I'd argue, because although, yeah, you can get that price, that's really fenced off for a lot of people, because in order to achieve that low price, typically you have to be a big genome center or a big university that can afford those machines that ultimately give you economy of scale. So the challenge that I'm working on, and many others, is saying, okay, how can you get rid of the cost that you need to start a genome center or maintain it? If you are at a university that doesn't typically do genomics, and so therefore you don't have anyone who even knows how to get started, how can we help educators not be isolated? How can we actually put them in touch and build their capacity so that they could do the sequencing? Also, if you're an educator, you still may not realize the full scope of, well, what are the potential things I could do with my students? What's valuable? What's interesting? What would they like to do? What would I like to do? And then as an educator, do I have enough time to teach these things and actually have a level of confidence? If you give an educator a new tool, their biggest fear is that their students are going to ask a question they don't know how to answer. And that delays them from putting it in front of the student sometimes, whereas the other attitude, which other educators also have sometimes, is let my students figure it out, and they'll tell me, and I'll work with them. Right? Those are different approaches which can work, and it makes it an equity issue. People may have seen this cartoon because it means that really some institutions are prepared to dip into the world of genomics, and others are still standing behind that fence, not quite to do, and so we need to get them the resources. So Nanopore has definitely been a way to do this very quickly for people who, like I said, I might assume, just based upon my work, that everybody thinks about sequencing all the time and knows all the details. But essentially, today, there are 4 major technologies that are in use for sequencing. The first one is the one that we use to sequence the human genome, Sanger sequencing. And it hasn't gone away in the way that, if you have a record player at home or an 8-track or something, just because a new technology comes, you still have it. It's still good for some things, but Sanger sequencing is one of the first technologies. It's what we call short read sequencing today, which does about 100 to 1,000 base pairs. But you can't really use it to sequence lots of things, but it's very accurate. And for small things you have questions about, we use this all the time with our students, where they might sequence a plant or try to identify whether the sushi is real. All those are examples. The biggest sequencing that you'll see widely deployed is Illumina, which, again, I'm not going to go into the deep details of it, but it allows you to generate millions of bases of DNA over the course of usually 2 days, 3 days. So this is the current standard. If you go to a hospital, very often that will be the ones that clinicians will use. The other two technologies are what we call long read. So long read allows you to sequence thousands, tens of thousands, hundreds, millions of base pairs. And actually, the human genome, the first draft of it was published in 2003, but it was never finished until 2022 about, depending on if you count when the paper was published versus the work, because there were always areas of the human genome they couldn't quite assemble. There were parts that were too repetitive and they couldn't, and it was Nanopore and PacBio, but really Nanopore, that allowed them to complete the genome by actually sequencing those pieces they couldn't quite reach. The key thing, if you look at the very last thing, is prices. Illumina is going to be tens of thousands to start up. I think that the smallest machines, right, you're talking about in the 60s, and then you're not talking about reagents and people, but Nanopore, you get started with 2,000. So that makes it really interesting. The way that Nanopore works is that literally in an embedded chip, you have a biological protein where you are putting a strand of DNA that's been prepared by some enzymatic process through that, and you're actually reading, it's basically a voltmeter, you're reading electrical signals. Oops, this is a touch. You're reading electrical signals in that pore that allow you to determine what the sequence is. And so that little device allows you to, or that nanopore, you get a readout, and what it really gives you the advantage of is in real time, in hours, I can start a process where I'm working with the sequencing. In the morning, I'm collecting DNA, but by the afternoon, I've got a sequence coming out. There are other advantages. I don't want to go too into like a molecular biology class here. One of the ones that's interesting is you can also do RNA. There's also people who are still working and getting to do protein. So it's something that, as a biologist, you could examine everything that's in our sort of toolkit of biology. Some of the ways that we've used it with high school students. So I was ready to do this just before the pandemic, and then the pandemic, so we kind of paused down. But this is just one example that we've done every summer. We have week-long summer camps, and so these are high school students. They are typically in the 11th or 12th grades, so the last 2 years of their high schooling. But starting the first year, they were sequencing some small aquatic plants, duckweed, which is being looked at as a biofuel. The next year, we partnered with some colleagues in Puerto Rico to sequence an endangered species, and so they were able to help generate a sequence for this particular Jamaican broom tree. The next year, the students went out into their garden or their backyard, and we collected chloroplasts, and they were able to do that. This year, we looked at soil, and they developed, over the course of 1 week, regular high school students. Now, I'm lying to you a little bit because they've probably taken some of our other courses before, so we didn't have to teach them how to pipette. They could use a computer. They maybe even know a little bit of Python. But the point is that we went from a bag of soil on Monday to a genome by Friday afternoon, right? So this is what a high school student, right? You guys have an undergraduate class that lasts a semester. At the undergraduate level, we can definitely do this. What we've been doing for a long time is preparing students with our programs to really do DNA sequencing, and in this case, this is another technique, DNA barcoding, where students can go out and, again, with a handful of really low-cost materials, ask the question about their environment around them, their food, whatever they like to look at. Sequencing was always the missing element here because we would have to send the samples away for sequencing. They could not, they'd have to wait 24, 48 hours to get the results back. But now what we're transitioning to is that the students can actually do the sequencing, so they can do the end-to-end workflow. Like some of the first students we did, this is a group of from public school students in the Bronx. They were looking at ginkgo, so they went into medicine shops, supplement shops, and they collected every single medicinal product, tea, that said that it was ginkgo because it's supposed to be good for memory or learning or your brain. But when they sequenced the DNA, all of the samples that were supposed to be ginkgo, I touch it again, none of them had ginkgo DNA. Like, none of them matched. the reference samples, what they did match was rice. So this is basically, you could imagine the rice paddy is gone and harvested, and they just take the rice clippings and grind those up. Because you have no idea when you're looking at a tablet, it's no longer visually identifiable. But that work was confirmed by the New York State Attorney General, and cease and desist letters were sent to the pharmacists because it was true, right? And they may not have even known that the sample that they were given wasn't really and authentically a capsule of ginkgo. So, but this is what high school students can do, and they love it, right? Because who would not want to be able to be? You want to be on the New York Times saying that you discovered this, right? So these are the types of discoveries that we're working, and Nanopore is going to power that even further. I have plans that are not on these slides, but this is the type of thing that students love it, because the gold standard for doing this type of education is that you don't know the outcome. The student can come up with their own question, but using a very common set of low-cost tools, they can explore thousands of different questions. So for the last 2 years, we've been working, this is a pilot project that's now ended, and we're ready. Oh, please, go ahead.
How do you stop the student from sequencing their own DNA?
Good question, right? So,
Or their friend's DNA.
or, for example, I mean, we also have things,
DNA.
we have things where we look at mitochondrial DNA, and so we look at mitochondrial specifically because we don't want to accidentally reveal paternity questions in the classroom, right? So in this case, particular things where we're doing an amplified product by PCR, we know what they're going to get because they can't get something that we don't want to look at. But in a more complicated nanopore procedure, one could take some of their own DNA and sequence it. Because we're working with the students, that's not really a real possibility. But in some of the larger rollouts of what we plan to do, you can actually bioinformatically exclude certain types of DNA from even appearing in the data set. So actually, the Nanopore software, there's a live sequencing component where it's actually blast searching the sequence that's coming back. And so you can make your analytic workflow specifically exclude human DNA so that you would never report that. And in a classroom environment, hey, yeah, accidentally, it could just be contamination, not even intentional. So I think our workflows would be the answer. Someone can get around that, but essentially it could be done. So we've been working with a group of, we now actually have about 100 educators across the US, where we have been working with them in a network to share materials, share protocols, discuss, get support in developing classroom activities. Our next thing would be to make that a bit bigger. This summer, we actually did 7 training workshops across the US. That's not including some of the other ones that were a little bit smaller. These are 3-day ones, but I was as far away as Guam. Just because I was there, I was going to go teach. That's about a little bit, same time zone as Melbourne. So on that side of the earth, that's a US territory. We had an NIH meeting, but I got to go all around to teach and to work with faculty educators who are tremendously excited for trying to do this. This is one of our workshops in New York. That was the year before. We were just starting to lift this off the ground. And switching gears here a little bit.
Sorry, can I ask you about Nanopore?
Absolutely.
I'm not familiar at all with
I saw you actually had it with you.
I have, no, this is my,
This is something else.
it's the same size.
Sorry, I thought something else.
No, it's the same size, and I probably actually have, I know I have a nanopore cable in here, I have one, so, but it's the same size. You had a question about it?
Yeah, no, I don't know anything about it, so I thought, is it a company? Well,
Yeah, so it's in Oxford Nanopore, so it's literally in Oxford. I was there yesterday visiting with them, and essentially it's been also about almost 20 years of development of this technology to go from an electronics experiment to basically say you can take an artificially designed protein, because naturally, I don't have those slides on this deck, but naturally there are microbes. I think it was an E. coli, but it's a bacterial microbe that you have a pore that they used as the model, and it's an artificial membrane that this pore is embedded in. So it creates this little cavity. What you don't really see very clearly here is that embedded in this is an electronic sensor. And so what happens is that as a piece of DNA is moved by a motor protein, it's unwound by basically a helicase case and pushed through, it disrupts the electrical current. And the fundamental principle of it is that essentially an A nucleotide disrupts that current slightly different than a T or a C or a G. And those really, really, really microvolt disruptions can actually be used by machine learning to interpret those changes in electrical signal and reconstruct what the original sequence could be. So it's a longer lecture to go deeper than that, but essentially...
I remember this a long time ago when
Yeah.
we were starting it. I think.
Well, now it's real. And it took a long time. So I started back in 2014 testing it, and it was starting to work, but it was nowhere near what you could bring into the classroom. And it's really only in the last, I would say, 4 or 5 years that you could start bringing it to the classroom. And I would say it's probably right now that I feel, yeah, you know what? I can put this in front of a high school teacher who's never done it, and I can get her in a couple days to be able to do it, even if she's not really, doesn't consider herself a molecular biologist. So it's easy enough now to use. It's not flawless. There will be mistakes, and you need a little bit of help, but it's actually quite doable. Yep. So let me change gears here. I think I'm working my way through my slides, trying to figure out what I wrote, and just broaden out to the final, let's say in the last 10 minutes, broaden out to some of the things that we've been working on. So I think you'll share the slide deck because you've got it uploaded to the system, or if you need a copy, Manny can help get it out. We've worked for a long time, and we have a number of papers where we have been thinking about the barriers that stop. This is an earlier one, I think, from 2019. We actually just published this in 2023 when we went back and just surveying teachers and looking at teachers' habits to determine what were the things and the support that they needed to bring the data component in, because the molecular biology, believe it or not, is kind of the easier side for most of the people that were trained in that area. The data is still an issue. And going back to the original theme that I had of education, one of the things, I think this is, last year we published a consensus paper, because one of our approaches to getting educators, but people of all sorts, up to speed is through the type of training that we deliver, what we call short format training workshops. When you go and you grab a group of people for a day or 2 or 3 to say, okay, you want to take a 3-day course of nanopore, or you want to take a 2-day course of bioinformatics, how can we improve that? So these papers, which I invite you to take a look at, I'll actually skip this one. These papers talk about sets of principles and recommendations that a whole group of people from really across the world came together to ask, like when we're designing short-term curricula for getting new skills out to people, because we said earlier, the problem is you're already a scientist who knows something, but there's just one more thing you're reaching for that you haven't had a chance to learn it yet. So you go and take a workshop, or you go take an exposure, but as it turns out, those workshops usually aren't as effective as you think, because they're not really well designed oftentimes to really realize what the limitations and the opportunities are of training. You'll have to look at the paper, but essentially we devised a set, this thing called the bicycle principles, a set of core principles of educational strategies that are quite proven, but which actually get lost when you try to do something in a short amount of time, versus if you have a whole semester and you can actually include them. So for one example, I'll just choose one just because I happen to know them all, but this particular one on effectiveness is about assessment. So if you take any university course, you're going to have formative and summative assessments built into that course, providing the student and you as a teacher feedback on whether what you're actually trying to train or teach is making it across. And the student can also know, hey, I'm doing well in this class, I'm not doing well. Hopefully the response is that, well, I need to study a little bit more. In a 3-day nanopore workshop, I could teach at you and yell at you for 3 days. And you may not really learn nanopore, or you may enjoy the food and the company and the coffee. But then when you go back to try it yourself, you realize that you're not It seemed to work before when I was doing it there, but I didn't understand as much, right? So what are the strategies, for example, for assessment, even in a short amount of time, that we make the argument that you should be obligated to do as a person who's doing these short workshops or short training sessions? How do you bake that in so that you actually provide evidence that the learning is actually happening? Or help the student, the learner in this case, place themselves to say, okay, I was able to capture this, but I still need to work harder to capture that. We know that 3 days is not going to be enough for almost any subject of technological depth, but at least we could start giving you strategies to learn on your own and know what parts you kind of got and what parts you need to focus on. That in itself is a whole talk, but the paper goes through it, including different principles that we talk about for taking training programs that exist and expanding them. There's a website that's attached to that called bikeprinciples.org. There's lectures, there's the paper itself, and there are communities of people working on these principles to think about how can we take the talent that we've already grown and move it to the next step of making it more effective by thinking about professional development for our educators. I also help run another community, Life-Sci Trainers, where people who do training across the world can come together for discussions or for exchange. My last gear change here, I guess I'm still with the bicycle in some ways, is one new project that I'm working on and happy to try to get off the ground and advertise, and we're hoping to open it next year. So this is an award that we got just last year. It seems a very long time ago, but there's always something new happening called Arecibo C3. So this is based in Puerto Rico, and it's a science center, sort of like what we're doing with the DNA, but a little bit more broad because we are incorporating STEM education research.
That's why you invited me to go, right?
Yes, I've already said that you've got to come down. Computation and community. Puerto Rico is a unique locale in the United States. It's in the Caribbean, which is always nice. And it's a predominantly Spanish-speaking territory. So my grandfather was from Guaynabo, just outside of San Juan in Puerto Rico. And so we have a mixture of STEM education research, community building, skill building, and it's a $5 million award with various institutions in Puerto Rico and the U.S. In some ways, it's a little bit like Cold Spring Harbor because it's a sort of isolated place. But what was formerly there, if you've seen like a James Bond or something, was this gigantic radio telescope that underwent a collapse a few years back. And so the property was still owned by the U.S. National Science Foundation, and they asked, well, what could we do? And so they thought, well, we can take it and still achieve the science mission of the site, even though it's not doing radio astronomy at the moment. There may be future plans, which I'm not allowed to talk about, so they don't tell me about it, but I know that they're working on other things. But at that site, what we're doing right now is working on renovations that will bring some new science teaching labs there. But it has a whole number of facilities, so there's on-site lodging for scientists to come for exchange. There's wonderful office space. This space right now is a temporary teaching lab, and we're working on exhibits and a science and visitor center. So it's going to be my latest project, dealing with all of these types of things and merging some of the ideas that we talked about. In fact, the very first pilot workshop that we held there this May was on nanopore sequencing, so we're excited about that. So I'm going to finish here, I guess, with a quote. That it's really this, re-education of people and development of STEM talent that's really, really important for us to do. So I want to thank the invitation for coffee and for everybody for listening to me for a few minutes. And happy to listen to you and learn from you. Anything that you found interested, I could talk about more. But I hope there might be something interesting there that you'll dig into and say, can we do this? Because you certainly can. And thank you very much for your attention.
So I'm going to put you in the spot, Liz. Why don't you tell us the amazing, and also Jimmy, the amazing stuff that you are doing so that he knows.
Yeah, I'd love to hear more.
Well, first, this was really
No, I
inspiring.
realized I wasted my life.
No.
I should
No.
have been doing something more useful like you.
No. It's all like, hey, this stuff is still ready for us to do lots of work.
But you mentioned in your slides, you
Yeah.
know, the scientists have become obsolete because their field has moved on, because that is exactly what's happened in our area, because so we work in MRI
Mhm.
and for measuring body composition.
Yep.
And we used to scan people and we would manually annotate the images. So
Yeah.
it took me like a day to work out how much fat somebody had.
Yeah.
But now we
Yeah.
manually train deep learning models, it's all
Yep.
machine learning, and we have clever young people who come from that background.
Exactly.
So while we give their work context,
Yes.
couldn't do any of it myself now. So
Correct.
I feel obsolete in my own areas. It's quite, that really resonated. Quite interested seeing it in writing. So.
Yeah. But it's also then a matter of like, how do you help people? Because the people who are experienced have such deep knowledge about the science and the context. So when they want to learn more, it's not saying that they now have to go off and completely retrain themselves as machine learning experts. But how do you take that interest that they do have in perhaps being a more effective interdisciplinary researcher by giving them the tools to now navigate more effectively with the person who does have that expertise and become a better team? How can we accelerate that? How can we make that effective?
But you think danger is that because you have these kind of like really clever young people
Mhm.
with all their machine learning doing the work, and as we eventually, you know, our deep knowledge, we all die out, who will replace us to give them the context? 'Cause
Well, that's very important because I think we saw this during the pandemic as a practical example of people who had maybe like best of intentions, expert deep mind worker at Facebook or something, and made discoveries that really weren't discoveries because they made no biological sense. They were just working with data without the context. So I think it's a matter of one, realizing that we need that context, but two, this is now veering off into my personal speculations, it means that we have to really rethink the need for professional development as professionals. It can't just be optional. It has to be built in that scientists, everyone has to say, hey, my skill set is only going to be this long before I really need to retrain. How do we change our academic approach, our academic, how would you say, perspective or mindset to say that we need to embed professional development, career spanning, it can't stop. And that's not to say, again, that everybody has to completely change what they're doing, but we need to realize the value of it and then make sure that the time spent bringing yourself into an updated area or even transitioning is very well spent because you probably still don't have that much time of it. And yeah, I can go on.
Because I thought, because you give a beautiful example of the 400 years for the telescope, the 40 years for the genomics, and I think it's probably been about 10 years, would you say, for the machine learning to imaging? Or maybe less? I guess as the pace of knowledge increases, the time
That you
required
have,
to
yeah.
skill
Yeah.
becomes smaller and smaller.
Well, that also means, for the people that do have that knowledge, how can they be made better educators, explainers, and teachers so that they're bringing people along? And also, how do they, as you build, literally, as you build software, making it easy and making it usable. Yeah, I don't need to learn how to write 10,000 lines of Python to use your thing. I just want my answer so that I can now ask better questions and not spend my whole day filling in. Right now, I could be made much more useful as long as you do your part and make your tool useful to somebody that's a non-expert. So it's collaboration. There's many questions. I see you waiting, and then I'll come back around.
I just want to see kind of like there are different things that it's a fantastic presentation. Thank you very much. Really eye-opening. I can think of many things to do. I mean, don't worry about these people become obsolete because we in modern life we think that we are we suffer the most, but in the English countryside about 600 years ago, someone invented a harvesting machine, and within 1 year, the whole population became obsolete.
Yeah.
So they took 40 years. You could become a pensioner in 40 years, so you don't have to worry about it.
Yeah.
So it's not. A long time ago, I'm a biochemist by training. I said that within 10 years, there are not going to be any labs, there are only going to be mathematicians. It just took 25 years instead of 10, but this is what happened.
Yeah.
Because technology, pipetting is going to become a machine thing. It's becoming more and more sort of skills are becoming less useful. Well, I think the power of something you've shown is this concept that people have been playing with about community science. Which people waffle on about, but the reality is they don't do community science. But this actually opens up the possibility to true community science, especially in areas of the country or inner cities where they don't have the technology or the capability to integrate. We have schools, open days when school children come, and we just show them things. They could stay at home and play a video game, would be far more interesting. But this opens up the possibility of doing actually real for them that they could actually follow up with school, and the link between higher education, universities, and secondary school, primary school become much more direct. Which I think is actually fascinating. I'm originally from Chile, and I could see how in the countryside in Chile you could actually apply the kind of stuff that you're doing in relation to the environment, new species and DNA sequencing that will resonate with young people, the environment, and so forth. I mean, I think it's absolutely fascinating. So I got so many questions I could go on and on. But when you say training, how much training do the students require to actually get to the point of use of the Nano4 system?
Yeah, I would say that if I had the students that's like a 9 to 4 p.m., so a whole day for a high school student, and a week is the minimum to get them like really started. And then you could say, now, okay, that's during the summer, but during the school year, maybe I can meet with you for a couple hours, on a Friday every week or something like that, or in the after-school club. So what we're trying to do is get students, but there are already students who are taking this and pretty independently saying, okay, I can do this myself. You give me some direction, because they're still in high school. But we have students like this, the endangered species. I worked with a high school student virtually over his school year, because he was with me in the summer physically to generate, but then over the school year, he had to learn some R. He had to learn how to install a package. But by the time we get to the end of it, he could actually assemble a genome and make a map, right? It's not perfect. There's much more to learn. But, as you said, the connection between the high school and the higher education means that the students that are really ready for it, you're giving them that opportunity. Because the average high school teacher is not going to have this background, so they can't necessarily go as deep. But if you have this link and you're supporting them, the high school teachers can do simpler experiments where they generate the interest, and then those students that are really dedicated can go to a next level. And to wrap up that thought, what you said about community science, right? I don't know what it is in the UK, but in the US, there's definitely surveys talking about public mistrust in science. And so I think the more that you have students doing this in their own community, right, that you create conversations. One of our mentoring programs has an element where the students are required to give back by conducting interviews with people and or scientists about current science-based topics. So this spring, our students had something about genetic diseases, and they interviewed Victoria Gray. We flew her up. She's the first patient cured of sickle cell using CRISPR technology. And so the high school students came up with their own questions based upon their research, and then they held a community roundtable that 100 people came to just talk about it, right? And so I think that during the pandemic, we did something similar where the students did interviews with NIH directors. But the point was, especially in communities where there aren't role model scientists, people don't come from generations of doctors or researchers, those students at the high school level are educated enough about biology that they can have a kitchen conversation about science. And I think that might be better received than someone in a lab coat or in a politician's suit telling them about science and what they think. So I think giving people that opportunity to do for themselves is empowering.
it's unbelievable how you've taken such an incredible complex kind of focus and made it so simple. Have there been any other industries that have approached you to actually see if they can implement, you know, that kind of system that you've built in their own like kind of unique
So
industry?
this is the work of many people. All of my colleagues at the Learning Center, our founder, who did this. But for example, with Oxford Nanopore, we have an MOU with them to actually basically lead their high school, their secondary education. So we make no money from them. I don't own any stock. But essentially, they realize that they can make the technology, but they don't have the scale to do education and training at the level that they're interested. They want it to be in the classroom. But we do that all the time. We have national and international networks. So what we are trying to do, and we're working with them, one, we have a very, very low-cost program. So since you're already using it, I saw you have a nanopore sequencer here. We have educational pricing that's like substantially less. So instead of a $600 kit, it's now $100. Instead of a $900 kit, it's $200. So we have stuff, so you can talk to me. But by reducing the price and then working with other educators. So there are a number of, the sort of excellent high school teachers who are actually high school teachers. Once they get good, they can bring their peers along. So we're doing train the trainer type things, so you can start to build enough capacity for it to become a ubiquitous deployment of that type of education at the high school level. So they're a company that we're working with to do that. Because it is, in that case, the technology is a key enabling factor. And then we provide the community infrastructure, because you need people to know about it and to feel supported as they're deploying it.
Because I can see this in so many industries, like explaining difficult complexity, and in terms of like someone that might be, like you say, like at a point in their job where they feel redundant,
Mm-hmm.
and taking that next leap. I mean, the whole process that you put together is you've done the most difficult thing, made it almost made it.
It's good to have some people specializing in parts of it that you don't have to do.
It's wonderful.
Namir, why don't you tell us a little bit about you? I'm putting up you on a spot, but I think he's actually an undergrad student.
Oh yeah, wonderful.
Yes, one of Dr. Coff's project students.
Oh.
Yes, we're doing pharmacogenomics projects at the moment, in
Uh-huh.
the final year. He invited me to come and hear you
Yeah.
speak.
Yeah.
So, what have you been working on? Give us a bit more details.
So looking at a specific CYP gene and looking at
It's like how to explain it. Where to start.
looking at the frequencies in different biogeographic groups and
Mhm.
biases maybe that, and then what effects that will have.
Mm-hmm. That's something that
Yeah.
we're interested in, because we also know with a lot of projects, we're not covering the diversity of the population, so we don't know the full story, right? And getting everybody involved is part of it. And something like this also, if people are not afraid and know the ins and outs of sequencing and how their data might be used, they might be more willing to participate.
Yes.
And they could actually give informed consents, not just checking a box,
Yeah, exactly.
but they understand what they're
Right.
doing when they're participating and what the benefit could be.
Yeah, they get benefits from that. They get better treatment. They get, yeah.
Yeah. Oh, excellent. That's great to hear. Yeah.
Thank you. Carmen, what are you doing here?
I get asked that question a lot.
I made you.
What are you doing here?
First of all, thank you so much. It
Thank
was
you
a
for
very
coming.
vision-expanding experience to listen to you. So what am I doing? I'm actually a pharmacist,
Mhm.
and I partially own a pharmacy in Turkey. So
Mhm.
I want to obviously improve myself and then improve the system in Turkey.
Mhm.
I actually have a question. I
Sure.
don't know the best or the right way to ask this.
To try.
Okay, so basically, do you think a trained pharmacist could basically like integrate the, was it nanopore?
Yeah, yeah.
The nanopore technology in a pharmacy just for like experimental reasons? Or
So I think the application of sequencing there's two. One is maybe a little bit not pharmacy based, although it's related. So I know that they're using it for HLA typing and transplant surgery, where you need to get sequence information, or you may not know these particular characteristics of the patient, and so you need to sequence, but it typically takes days, but you can do it now in hours. But when you talk about things like pharmacogenomics, where you're actually saying, hey, how will this patient react? And it's an interesting thing. I don't know if people have quite proposed it this way, but I was talking to many about the poor state of precision medicine in many places, including the US, where we're learning more and more about the genome and how a patient might react based upon their unique genetic background to a drug. Certainly an effort is focused upon doctors to, let's say, order the test to say, hey, we know, for example, things like blood thinners, where there's very, very specific responses that different patients will have, and you may not know in advance to a blood thinner what will happen. But pharmacy is another layer of, could that be a test that when you go in for your vaccine, or when you go to pick up a drug, they're like, oh, this has a very specific pharmacogenetic profile. We'd like to do a genetic test on you. Can you wait an hour? And then could you actually sample, right? Or especially something, probably this is a drug that they're going to be on for months, or if not years, or if not, wouldn't it be great when you're picking up that prescription to have that profile, so that your pharmacist could actually say, your doctor has ordered this dosage, but actually based upon this is the recommended dosage. We'll have a consult. So certainly at that pharmaceutical layer, there should be the idea of genetic testing, and also as we learn more about different populations, all of our testing may be masking what really is the effect of a particular background, even as we sort of also untangle genetics from race. So people might think, well, you are this background, or even somebody might say they're Turkish, but that may not tell you enough about their genetics to actually say that you're likely or unlikely to respond to a particular treatment. So there certainly is pharmaceutical applications that are not fully tapped, let's say.
Thank you.
So I like. So I'm very much aware of sort of the diversity efforts that are happening on this side of the Atlantic. But I would be quite interested in understanding how, for instance, the All of Us initiative, and in general, how are sort of perceptions and the way science is being done in the US trying to address the disparities that we currently have on data?
There's many, many different efforts, and they're all in different, it's very different contexts, right? In some ways, we talked about UK Biobank is probably sort of the gold standard of practice. The US is going to be more complicated. There are lots of different efforts. There are lots of people who care. I think that there could be more done with outreach in terms of people's perceptions and understanding of what are the benefits. There are also really different communities have different thoughts on how they would like to interact with genomics or not. So a couple of weeks ago at Cold Spring Harbor, we had an Indigenous Reference Genome meeting organized by a friend of mine who's one of a handful of Native American Navajo geneticists and genomicists, because they did not exist before. It's taken that long for the training to reach that community, so that within the community they have their own scientists. But as a rule, the Navajo Nation does not participate in many of these things, because in the past they thought that their participation has been inequitable, and largely sequencing or different activities have been done that extracted value from their DNA, because now you found a new drug target. But then, even if a pharmaceutical was produced, now they can't afford that pharmaceutical, so the benefit was not returned.
Can you put me in touch with him?
I'm going to be putting you in touch with a lot of people.
Seriously?
But so, and they run, so the person I was talking about is actually her, Crystal, but her colleague she's part of the same effort, but her colleague Joseph Aracheta, they run the first Native American biobank on the reservation, because by being on the reservation, they actually have a whole different set. Tribal law overrules the local law, and so that they can have protection for their data. Also, there were a number of groups from New Zealand, University of Aotearoa, talking about what is the Maori position to not only personal human genomics, but also biodiversity genomics. So all of these different things are coming into crystallization. It's going to become more important as there's more capacity to do genomic sequencing. And anyone, like you said, if somebody decides they want to do something that maybe they really shouldn't, how do we become sophisticated in understanding of where the ethical boundaries are, and then take up the appropriate actions to manage any risks that are there?
Yeah, I mean, this discussion going on for quite a while on biopiracy, which is, big farmers used to go to all these local villages, just take all the knowledge,
Correct.
extract them, and run.
Correct.
And that starts from the pill
Correct.
for women not to get pregnant, which was actually
And
taken from,
in my particular role,
Mexican women.
yeah, in my particular role, there's not all that much that I could do about it.
No.
But the more important thing that I can do about it is I could even just creating that awareness from a very early age that, hey, you need to handle DNA responsibly. Here's why. And you hope that one day that high school student, when they become a PhD student and then are working on a genomics process, have in the back of their head some of those ethical guidelines, which usually don't get taught to the very end of the education, that they're actually from the beginning aware that I need to act responsibly.
Yeah, I think one of the way to stop biopiracy is to empower the local people to have rather than fight the technology.
Correct.
to
Have the
actually
technology.
have
Be
the
able to do
knowledge
it.
To be able to use it themselves.
Correct.
According to
Absolutely.
Very fascinating.
If you were to justify why, say for instance Peru, why,
Just for random.
for instance, for random reasons. So why should they do the analysis there? Why should they do the the sequencing and rather than somewhere else? If you were going to convince a politician
Sure.
that that's the right thing, what
So
would you say?
I think one of there are a number of papers on what's called helicopter science of research groups that have the ability to do sequencing, analyze the data, have decades of expertise, right, coming into a community or into a country and essentially gobbling up, right, doing the experiments, doing the analyses, taking information, not involving any of the local scientists, not building any capacity, and then they publish their paper and generate either academic benefit or potentially a financial benefit somewhere else. So for individual groups and nations to generate that capacity is extremely important. And really until recently, like within the last 5 years, right, we know that long read technology like PacBio or Nanopore is even more valuable in some cases for doing the genomics. But a PacBio machine, Revio, is $900,000 to get started. The facility in Nigeria, they're building a whole new campus for $900,000 that they have, right, versus one machine. That then you need a specialized person to run, that you need this. What we're offering is saying that Nanopore is the current only option for this type of thing. Hey, there are going to be other companies who do something better. I will work with them. But at the moment, that allows for $2,000, $3,000, and a high school student to get started doing that locally and then protecting the intellectual and data rights and potentially patent rights to things in their community. So there's a gigantic case to be made for local capacity building. Yeah.
Thank you.
I'm at the top of the hour. I have a quick call that I have to, but if you're still around, then I could still have coffee with you. What's your plan? Good?
That's all, folks.
Good.
Absolutely brilliant.
Thank you so much for giving me an audience.
Oh,
I'm sure there'll be more future conversations. Oh, yeah, I hope we recorded.